I'm going to discuss concentration independent and concentration dependent models. And I'm going to use for this examples from diet reconstruction problems. Of course, the principles behind what I'm about to discuss are also applicable to non-diet reconstruction problems. So let me start with the following example, in which we have one consumer and three different food groups. Plant foods, terrestrial animals, this would be the meat of terrestrial animals and the meat from fish. And the question that I have is, what is the contribution of the protein within each food group towards the consumer? Notice that this is different from asking what is the contribution of each food group as a whole towards the consumer, because I'm only interested in the protein fraction. So this could be defined as a concentration independent model. And one often sees these type of models applied to diet reconstruction problems in archaeology or ecology. And typically two isotopic ratios are measured, carbon and nitrogen isotopic ratios measured in the protein of the different food groups and in the consumer. Now remember that the question that we are asking is what is the protein contribution towards the consumer? But by measuring these two isotopes, well, what one truly would have to consider as concentrations, if one would have to consider concentrations, would be those of the elements carbon and nitrogen within the protein. So how come one is obtaining estimates on the contribution of protein towards the consumer if these concentrations are the ones that one would have to consider? Well, there are a couple of implicit assumptions here. One is that the protein content among the different food groups is proportional to the carbon content. And two, that the protein content is proportional to the nitrogen content among the different food groups. And these two statements have as a logical consequence that the carbon content among the different food groups is proportional to the nitrogen content. Now, this is not strictly true. If you look at the nitrogen and carbon compositions of different proteins, they can vary, but they vary slightly. So this is approximately true. And this is why one can employ these concentration-independent models to obtain estimates on the protein contribution from different foods. Let's take the following example, in which we have two foods, food A and food B, and the carbon to nitrogen proportions vary among the two food groups. In this case, it would be one would have to necessarily to employ a concentration-dependent model. And if our question remains what is the contribution in terms of protein towards the consumer, then one would have to express these concentrations of carbon and nitrogen relative to the protein. If our question is what is the contribution of food as a whole, then one would have to express the concentrations of carbon and nitrogen relative to the whole food. But let's assume that this is not the case. We return to our previous problem. And indeed, the carbon to nitrogen proportion or ratio is approximately the same for the different food groups. How does one implement this problem in fruits? Well, fruits allows you to do this in two ways. Well, the first obvious one is that you would consider two fractions. The carbon, elemental carbon within the protein, and the elemental nitrogen within the protein. But because these two, because the concentrations of these two elements are proportional among the different food groups, you could also employ just one simple fraction that you could call protein. And this protein is associated to the two isotopic signals, carbon and nitrogen. Okay? So let me show you how in practice this would work in fruits. So here I have the two models with just one fraction and with two fractions. Okay, just a second here, I need to close these windows. I'm going to open here again with one fraction and with two fractions. Okay. So this is the one. Okay, do this so that we can compare them closely. Okay, and okay. So in this we have the two fractions, and I call them fraction carbon and fraction nitrogen. And in this model we have the one fraction that I call protein. When we look at the offsets and weights in the model with the two fractions, I have to inform fruits that the C13 signal measured in the consumer is 100% determined by the fraction carbon, and that the N15 signal measured in the consumer is 100% determined by the fraction nitrogen. In the model with just one fraction, we have that the C13 and N15 signals measured in the consumer are 100% derived from the protein. And this is valid because we have this proportionality relative to carbon and nitrogen. Okay. The way one defines the food values will be slightly different. Well, will be different in the two models. So let's take the easy one first with just one fraction. So we have the C13 and N15 signals for the protein fraction within each food group defined here. But in the model with two fractions, well, we have these black cells here which we cannot edit, and it's basically saying that the plant, for example, the plant carbon fraction, well, the only signal that we have to define here is the carbon isotopic signal and its uncertainty, because this fraction will not contribute to N15. And these cells are in black because we informed the model that this fraction carbon does not contribute to the N15 signal. But the fraction nitrogen in plants does contribute to the nitrogen isotopic signal, and this is why these cells are white and one can edit them, but it doesn't contribute to the carbon isotopic signal, and this is why these ones are black and one cannot edit them. Okay. Concentrations. It's a concentration independent model. These values are all set to 100 and it's not possible to edit them, and the same thing here with just one fraction. So let's look at the estimates in each case, and let's see that both models, quite naturally, of course, necessarily give the same estimates. So we have here with the two fractions, and now I'm going to generate the estimates for the model with just one fraction, and we just need to wait 1 second, and then we can compare the box plots, and here they are, and as you can see, the estimates are the same. If one looks at the results report and the main numeric estimates, you will notice that these are essentially the same. There's a non-significant difference between the two of below 1%, and this has to do with the fact that the implementation of both models is slightly different. Now, coming back to the slide that I showed you, so as I just mentioned, you have two ways of implementing this concentration independent model in fruits, by considering either two fractions or just one fraction. So what would be the advantage of considering just one fraction? Well, one immediate thing I could say is that, as you probably noticed, it's a bit simpler to implement the one fraction option. But truly the advantages behind defining this protein fraction come with concentration-dependent models. So let's no longer have as our question the protein source, but the contribution of the foods as a whole towards our consumer. This becomes a concentration-dependent model in which one has to consider the concentration of the protein within each food group. And this protein determines our carbon and nitrogen isotopic signals in the consumer. But one could consider additional fractions, for example, carbohydrates or lipids within each food group. And in fruits you can actually define additional fractions that do not contribute to any isotopic signal. But the fact that you define additional fractions within fruits means that you will obtain estimates on the relative contributions of the different fractions. To put this in a simple way, one would obtain as an estimate the levels of protein intake by the consumer, and this could be an estimate which is of interest. This would also allow us to implement priors that establish relationships among these two fractions. So, for example, for certain consumers, there are restrictions on the allowed levels of protein intake. So it could be that the amounts of protein in diet are restricted within a certain interval. And by defining these two fractions, one could define what is the interval within which protein intake is acceptable. And this could lead to more precise or even accurate estimates. Okay, so the advantages are truly seen when one has a concentration-dependent model. Are there circumstances in which one is imposed the use of concentration-dependent models? Well, let's consider the following. That we have before our protein fraction, but now we are using an additional proxy measured in the consumer, the sulfur isotopic signal, which of course will be determined by the sulfur fraction within the protein of each food group. And if you look at typical concentrations of sulfur-bearing amino acids in protein, you will notice that for food groups such as the meat of terrestrial animals or fish, these concentration, the proportions of sulfur-bearing amino acids in the protein of terrestrial animals and fish is typically much higher than the values observed in plants. And so we don't have this assumption that the relationship between the concentrations of sulfur versus protein is the same among the different food groups, and so this necessarily implies that a concentration-dependent model needs to be employed. And that's it.